Hello students, welcome to EPG Pathshala. I am Dr. Rashida Kanchwala. Today I am going to talk about water soluble pigments. Students, vegetables and fruits are important ingredients of our diet. All of you must have rejoiced them. The beautiful color and flavor of fruits and vegetables are deriving factors towards them. Today, I am going to teach you about different water soluble pigments. After going through this module, you will be able to know properties of various water soluble pigments, gain an overview of the bioavailability and biological functions of a range of water soluble pigments, to recognize the beneficial actions of water soluble pigments against various pathological conditions in plants and human beings. Plants produce more than 2 lakh different types of compounds. Many of them are pigmented. The color of plant can be recognized by perceiving reflected or transmitted light. As we perceive lights of wavelengths between 380 and 730 nanometer only. Pigments are the chemicals produced by plants which assist in photosynthesis, pollination and protecting them from oxidative stress. Pigments of plants are present in different bodies and forms. They may be present in vacuoles, plastids or as crystals dispersed in the protoplasm of the cells. Vacuoles are the separate compartments in the cell cytoplasm. They are filled with a fluid, cell sap, and are composed of dissolved sugars, salts, organic acids, pigments and other materials. Pigments may be present in vacuoles or macroscopic water cell in the plant cells. Anthocyanins and some other flavonoids and battalions are vacuolar pigments as they are present in water sacs. So they are water soluble. Plastids are located within the cytoplasm. They are separate inclusion bodies. In most of the pigments occurs in these plastids. A pigment may be present in the protoplasm as crystal. The structure of pigments are complex and as per presence or absence of hydrophilic and hydrophobic chains, pigments can be divided into two categories, that is fat soluble and water soluble. Here we will discuss water soluble pigments. The chief water soluble pigments are flavonoids, battalions, phycobilins, allophycocyanes and some others. Let us learn about them one by one. First of all, we will discuss flavonoids. Flavonoids are widely distributed plant pigments. Every type of plant contains one or more of these pigments in different chemical formulations. They are found in the epidermal tissues of flowers and fruits, where they are present in the vacuolar shape of vacuoles. Therefore, they are also termed as vacuolar pigments. Flavonoids are also synthesized as intermediary products during light absorption by phytochrome and flavins. Anthocyanins are the most common class of flavonoids. As a dietary component, flavonoids are thought to have health properties due to their high antioxidant capacity. Flavanols have ability to induce human protective enzyme system. Flavonoids are responsible for secondary antioxidant defense system in plant tissues where they are exposed to different abiotic and biotic stresses. Flavonoids are a large family of over 5,000 hydroxylated polyphenolic compounds. Chemically, they have the general structure of 15 carbon skeleton, which consists of 2 phenyl rings A and B and a 3 carbon bridge. In most of the flavonoids, the 3 carbon bridge is condensed through an oxygen into a heterocyclic ring termed as ring C. According to the IUPAC nomenclature, this carbon structure can be abbreviated C6, C3, C6. Let we be acquainted with their classes. All flavonoids contain 2 phenyl rings A and B and a 3 carbon bridge. In most of the flavonoids, the 3 carbon bridge is condensed through an oxygen into a heterocyclic ring C. The oxidation pattern of C ring differs, and accordingly there are 14 major types of flavonoids. Their activities are structure dependent. The types of flavonoids differ as per structural class, degree of hydroxylation, substitution and conjugation, and degree of polymerization. Functional hydroxyl groups in flavonoids mediate their antioxidant effects by scavenging free radicals and or by chelating metal ions. Let me discuss derivatives of flavonoids. Flavonoids occur as aglycones, glycosides and methylated derivatives. The basic flavonoid structure is aglycone. 6-member ring condensed with the benzene ring is either alpha pyrrone or the dihydro derivative of alpha pyrrone. The position of the benzoate substituent divides the flavonoid class into flavonoids which have benzoate at second position, isoflavonoid which has it at third position, and the difference between flavanols and flavanol is at C2 and C3, where there is a hydroxyl group at the third position and a C2-C3 double bond in flavonols. Aglycones are generally more active than glycosides. Methylated compounds of flavonoids are generally more biologically active than non-methylated compounds. Methylation and glyconation or glycolation requires specific methyl transferase or glycosyl transferase enzyme. Methyl ethers and acetyl ethers of the alcohol groups are now known to occur in nature. Thus, flavonoids can chiefly be classified into flavanols, flavanone, flavanols, flavanones, flavinoids and its isoflavonoids. Let we learn about flavons. Flavons have a double bond between position 2 and 3 and a ketone in position 4 of the C ring. Most flavons of vegetables and fruits has a hydroxyl group in position 5 of the A ring, while the hydroxylation in other position for the most part in position 7 of the A ring or 3 or 4 of the B ring. Hydroxylation may vary according to the taxonomic classification of the particular vegetable or fruit. They are generally glycolated. Glycolation occurs primarily on position 5 and 7. They may be methylated or acylated. Methylation and acylation occurs on the hydroxyl groups of the B ring. Some flavones such as nobilatin and tangeratin are polyhydroxylated. There are 5 common flavones and flavanols. They are epigenin, luteolin, quercetin, camphorol and myrcetin. Now we come to flavanol. Compared to flavanols, they have a hydroxyl group in position 3 of the C ring, which may also be glycolylated. Flavonols are very diverse in methylation and hydroxylation patterns. If different glycylation patterns of flavonols are considered, they are perhaps the most common and largest subgroups of flavonoids in fruits and vegetables. For example, quercetin is present in many plant foods. Flavonols may be present as such or as glycosides in nature. The another flavonoid, flavonols, which are also called as dihydroflavonones, have the C ring saturated. Therefore, unlike flavons, the double bond between position 2 and 3 is saturated, and this is the only structural difference between the two subgroups of flavonoids. Therefore, they are given a nomenclature of 2,3-dihydro 2-phenylchromane 4,1 as per IUPAC nomenclature. The flavonols can be multi-hydroxylated and several hydroxyl groups can be glycosylated and or methylated. Some have unique patterns of substitution due to which this group contains a great number of substituted derivatives. Flavonols are present in many herbs and fruits, but among all the plant kingdom, the species where flavonols are most commonly found are citrus. Citrus contains a significantly big amount of flavonols, being the richest source of flavonols, especially when they are still immature. Let me come to flavanols. Flavanols, also called dihydroflavanols, are the 3-hydroxy derivatives of flavonols. They are highly diversified and multi-substituted subgroups. They are 3-hydroxy 2,3-dihydro 2-phenylchromane 4,1 as per IUPAC nomenclature. Let me discuss flavanols or flavan-3-ols or catechins. In flavanols, the hydroxyl group is almost always bound to position 3 of C ring. Therefore, they are also referred to flavan-3-ols. They are called catechins as well. They are also saturated at position 2 and 3, whereas most of the flavonoids have a double bond between position 2 and 3. Flavanols have 2 chiral centers in the molecules on position 2 and 3, due to which they form 4 distereoisomer. Their cis and trans isomers are epicatechin and catechin. Epicatechin is the isomer with the cis configuration and catechin is the one with the trans configuration. Let me come to polymers of flavanols. An important feature of flavanols, particularly of catechin and epicatechin, is their ability to form polymers. They form two polymers which are termed as proanthocyanidins and contains tannins. Proanthocyanidins, as the name implies, are precursors of anthocyanidins, which are formed from acid catalyzed cleavage of proanthocyanidin. Proanthocyanidins contain 2 to 60 monomers or flavanols. Monomeric and oligomeric flavanols are strong antioxidants. Flavonoids are found in many fruits, particularly in the skins of grapes, apple and blueberries. Monomeric flavonoids, catechin and epicatechin and their derivatives are the major flavonoids in tea leaves and cocoa beans. Let us discuss isoflavonoids. Isoflavonoids are derived from flavonoid biosynthesis pathway, while flavonoids have the 2-phenylchromane 4,1 backbone. Isoflavonoids have the 3-phenyl chromane 4-1 big bond. Their chemical formula is C15H10O2. They have no hydroxyl group substitution at position 2. Isoflavonoids are a class of flavonoids which are biologically active. As many isoflavonoid compounds have biological effects by the estrogen receptor, isoflavonoids and derivatives are sometimes referred to as phytoestrogens. The isoflavonoid group is broad and includes many structurally similar groups, including isoflavones, isoflavonols, and isoflavins. Pterocarbons. Let we know isoflavones. Isoflavones differ from flavones in location of the phenyl group. In our food, the chief source of isoflavones are soybeans. The major isoflavone in soybean are genistein and dedzin. Isoflavones and their derivatives work as phytoalexin compounds. The compounds which wards off diseases causing pathogens and microbes. Soybean uses isoflavones to stimulate soil microrhizobium to form nitrogen-fixing root nodules. In soybean, isoflavones are present as glycosides. Fermentation or digestion of soybean or soya products results in the release of the sugar molecule from the isoflavone and glycosides, leaving an isoflavone aglycone. Soya isoflavone glycosides are called genistein, diazine, and glycitin. Let me discuss pterocarpans. Pterocarpans are derivatives of isoflavonoids found in the legume, seed, or bean family. It is a group of compounds which can be described as benzo-pyreno-furanobenzene. They generally bear structural resemblance to the isoflavonoids that possess antibiotic activity and are produced by plants tissues in response to infection. Now we come to anthocyanidins. Anthocyanidins are the only pigment of flavonoid group which gives color to plants. They are flavillium cations and are generally present as chloride salts. They are the only group of flavonoids that gives plants colors. All other flavonoids are colorless. Anthocyanins are glycosides of anthocyanidins. Sugar units are bound mostly to position 3 of the C ring and they are often conjugated with phenolic acids such as ferulic acid. They may be present as mono or diglycosides. Different glycosidic combinations of anthocyanidins are 3 monoglycosides, 5 diglycosides, 3 diglycosides, 5 monoglycosides and 7 monoglycosides. The most common sugars present in these natural pigments is glucose, which is the most common sugar. Besides this, fructose, galactose, xylose, arabinose and rhamnose may also be present. There are a variety of anthocyanidins in plants as per number and position of hydroxyl and methoxyl groups. 6 of them are commonly found in vegetables and fruits. They are pelargonidin, cyanidin, delphinidin, tunidin and peonidin and malvinidin. Let us see the effect of different factors on color of anthocyanins. The color of anthocyanins depends on the pH and also by methylation or acylation of the hydroxyl groups on the A and B rings. Different derivatives of anthocyanins behave differently at a given pH and their resultant value is the result of difference in hue of different fruits containing anthocyanins. In this respect, example of 2 anthocyanins are worth citable. Pelargonidin is generally orange red and delphinidin is bluish red in acidic pH. Change in pH from acid to base results in brewing of both of these pigments. Two vegetables or fruits may have same pigment but different in their pH may result in different hues. Example of red rose and blue cornflower is words citable in this regard, both of which have same signing but acidic pH of rose gives a red hue and basic pH of cornflower provides a blue hue. It has been found that a particular cyanidin may give different hue in different concentrations. It has been reported that delphinidin solution of different concentrations provides different hues. A blue, purple or red hue may be reported from dilute, intermediate or concentrated solutions of delphinidin. Anthocyanins are generally present as more than one derivatives. Different fruits and vegetables have difference in this mixture composition which results in varied hues in them. Presence of other flavonoids not only contain different anthocyanins but they also contain different flavonoids along with them. Presence of anthocyanins, carotenoids and tannins provides different hues to different vegetations. Let me know about chalcones. Chalcones and dihydrochalcones are flavonoids with open structure. Anthocyanins form chalcones at higher pH, that is in basic conditions, and they are intermediary products in flavonoid biosynthesis. They are colorless. These are natural products which belong to flavonoid and are widely spread in plants. The existence of alpha-beta unsaturated ketone moiety in chalcones is a common part found in a large number of biologically active compounds. The open ring chalcones are found in fruits such as apples and hops. Now it is the time to know about physiological role of flavonoids. Flavonoids attract the insects for pollination. They absorb the heat and it increases the transpiration in cell. They increases osmotic pressure in the cell and contributes to the food flavor and colors. Now, let me discuss Battalions. Battalions are found in plants of the caryophyllales. They are a class of red and yellow indole-derived pigments which replace anthocyanin pigments. They are most often noticeable in the petals of flowers, but they may color fruits, petal, leaves and other parts of the plant. They include pigments such as those found in beets. The name Battalion comes from the Latin name of the common beets from which Battalions were first extracted. The deep red color of beets, bougainvillea, amaranth and many cactuses results from the presence of Battalion. The particular shades of Battalions is red to purple, which is its distinctive shade. It is different from anthocyanin pigment found in most plants. Two pigments, betanin and valgaxanthine, belong to the betanin group of colors. These colors do not only exist in beetroot but also in several other plants and mushrooms. Except beets, none of these are important as food sources. Betanins are classified on their structure and divided into two groups, beta-cyanin and beta-xanthines, with red, purple and yellow colors respectively. More than 50 betalains have been described. There are two categories of betalains, beta-cyanin, which includes the reddish to violet betalan pigments. Many types of beta-cyanins are present in plants. Chief of them are betanin, isobetaine, probetaine and neobetaine. Betalains are comparatively more stable at acidic pH than basic ones. They are stable between pH 3 and 7 but at maximum color stability at pH 5.5 but are susceptible toward light and temperature. Beta-xanthines are yellow to orange betalene pigments. The chief beta-xanthines in plants are vulgaxanthines, miraxanthin, portulaxanthin and indicaxanthin. Beta-xanthin simulates with anthocyanin, therefore they were first related to them. Researchers revealed them as separate entities. They have fungicidal properties. Although both betalains and anthocyanins are water-soluble vacuolar pigments, but both of them are chemically different. It has also been found that both of them do not coexist. In a plant, there may either be betaline or anthocyanin. Betalains contain nitrogen, whereas anthocyanins do not contain nitrogen. Betalains are aromatic indole derivatives synthesized from tyrosine. Therefore, they are not related chemically also with anthocyanins or flavonoids. They are glycosides having a sugar and a colored portion which gets hydrolyzed into the sugar glucose betanidine. Their synthesis is promoted by light. Let we come to phycobilins. Phycobilins are water-soluble photosynthetic pigments. They are not present in higher plants but do occur in red algae and the cyanobacteria, a group of photosynthetic bacteria. It is a protein from the light harvesting phycobiliprotein family. Phycocyanin, phycoerythrin and phycoerythrocyanin are other members of the family. They are accessory pigments to chlorophyll. All phycobiliproteins are water soluble and are aggregates to form cluster that adheres to the membrane. The cluster of phycobiliproteins are known as phycobilisomes. Allophycocin absorbs and emits red light between range of 615 and 660 nm and is really found in cyanobacteria, also called blue-green algae and red algae. Phycobilin pigment have fluorescent properties that are used in immunoassay kits. So we can sum up that colors in plants are due to some pigments within their cells. The pigments may be soluble in water or fat. Their behavior is determined as by their solubility. Most of the water soluble pigments are affected by heat, light and pH changes. Therefore, vegetables containing them should be handled as per their behavior. Most of them are accessory photosynthetic pigments. Along with this, they are antioxidants and they are important constituents for sustainability of plants and health of humans. Thank you.